Assalamualaikum and hi everyone. So back in this video, we are going to learn more about what happened after transcription. So now I want you to turn your bio notes to page 127. Okay, so basically in transcription, we have learned that we want to produce a pre-mRNA. Okay, remember it's a pre-mRNA, bermaksud dia belum lagi matang. Macam mana untuk kita mematangkan mRNA itu? Itulah yang kita akan belajar hari ini. So kita tengok sini. Jadi kita akan belajar tentang RNA processing. Okay, so RNA processing ini adalah satu proses di mana kita modify two ends of a eukaryotic pre-mRNA molecule. So maksudnya ini kita dapat dari transcription. Ini hasil daripada transcription. So I'm going to put here transcription product. So now you have to know the primary mRNA transcript. Kita akan mempersiapsiagakan dia untuk apa? So we learn from central dogma, it is from transcription to translation. Okay, so before, in between transcription dengan translation kan, kita akan ada RNA processing. Okay, RNA processing ada dua step. I hope you guys remember from our online lecture. The first one is alteration of mRNA ends. Alteration bermaksud mengubah. Okay, mengubah. Alter kan, alter mengubah. Contoh alter baju kamu terlampau besar, mau bagi kecil, so kita ubahlah baju tu mengikut saiz kamu. Okay, so in the case of mRNA, kita akan ubah ke kedua-dua hujung mRNA. So we will take a look at here. Kita sudah belajar dekat 5' sini kita akan tambah 5' cap. So that's why here 5' and receive a 5' cap. Okay, so ini 5, mestilah di hujungnya ada 3'. So 3' dekat sini kita akan tambah poly A tel. Okay, dekat online lecture hari tu Miss sudah cakap poly bermaksud banyak, many, like a lot of A. And you can see here how many A's do we add at the end of 3N. 50 to 350 adenine nucleotide. So that's a lot of nucleotide. Alright, so when you look at 3'N, you will realize kita ada satu codon di sini. Bukan codon, kita panggil sebagai signal. Do you guys remember the signal? So during termination, the transcribed pre-mRNA will have this signal. So we call this as polyadenylation signal. AUA kan? So this is 5' dash AU dash 3'. So this is the polyadenylation signal. So bila ada transcribe signal ini, transcription stop. Okay, that's why this is the end of it. Dan dekat 3N kita tambah pula dengan poly A tel. Okay, boleh? So you have to know what's the function of it. Soalan pernah keluar lah juga, dia tanya kenapa. What's the importance? So we actually want to promote the export of mRNA from nucleus. So we know for translation, the mRNA will leave nucleus. I keep saying before, kampung nucleus, dia akan ke bandar. Bandar mana? Bandar cytoplasm. So this is actually for translation. For translation in cytoplasm. Okay, so that's the first importance. The second one, what's the function? Help to protect mRNA from degradation. So basically, when mRNA kan keluar daripada nukleus, dekat cytoplasm tu terlalu banyak enzim. So the enzyme actually can degrade the mRNA. Degrade bermaksud memusnahkan mRNA itu. Kita nak mau dia musnah. That's why lah kita tambah keselamatan di sini. Kita tambah 5' cap dekat 5N and we add poly A tel at the 3N. Okay? And the last one, help ribosome to attach to 5N of mRNA in cytoplasm. So this has to do with initiation stage in translation. Next video Miss akan ajar. Okay, so this is all about the first one, alteration of mRNA ends. So let's take a look the second step. Second step kita panggil sebagai split genes and RNA splicing. So let's take a look here. So dekat sini kita tahu ada sudah 5' cap. Okay, ada sudah warna kuning. Poly A tel pun sudah ada. But now we want to remove whatever not important anymore. So kita sudah belajar juga intron. Okay, so maksudnya intron ini 1, 2. This is the non-coding segment. So non-coding bermaksud dia tidak ada membawa apa-apa coding yang penting dalam badan kita. So usually when we do not want it, we will remove it. So that's what happening. Segmen ini tidak diperlukan untuk proses seterusnya iaitu translation. So kita buang lah sebab dia tak penting. Okay, so intron tak penting, mestilah lagi satu exon penting. So exon adalah coding segment. Coding segment ini diperlukan sebab inilah kod-kod yang diperlukan untuk buat protein kan, untuk buat amino acid nanti dalam proses translation. So this is dia akan remain dekat mature mRNA. Okay, in case kamu nak dapat bayangkan macam mana kita remove satu protein dan juga small RNA bernama spliceosome, dia akan remove and then dia akan join. Okay, lagi sekali Miss ulang. Spliceosome ini, dia akan cut the intron, which is the non-coding segment, and then it will join the exon together. Okay, so can you imagine it? So this is what's happening. Introns are removed. So this part remove, kita nak mau kan? So ini kita remove and then exons are joined together. Okay, so that's what happened during RNA splicing. Sebab tu nama dia split gene. Kita akan split dahulu, kita buang intron, dan RNA splicing akan dijalankan oleh spliceosome. So now mRNA kita sudah matang. Dalam biologi kita panggil dia sebagai mature mRNA. So you guys can put it here. This is mature mRNA. Okay, so that's all for this video. Let's try to recall. Before we do translation, which is after transcription, we have to process the RNA. So we call it as RNA processing. RNA processing ada dua step. Satu, kita alteration of mRNA ends, di mana kita tambah 5' cap dekat 5' end. And then we add polyethyl to 3' end. So that's the first one. The second one, we will do split genes and RNA splicing, where we will remove intron and we will join together the exon by using spliceosome. And now we will have the mature mRNA. So I hope this video helps and I'll see you in the next one. Bye!